Hi, everyone. Welcome to our Monty Hart lecture series. It's a great pleasure to have today Dr. Sean Heffron that will be speaking to us about excess body weight, atherosclerosis, and inflammation resolution. Dr. Heffron is an assistant professor at NYU. He did his medical school at Duke, then went to MGH to do internal medicine and back to New York for his cardiology fellowship at NYU, where he stayed as an attending and where he's now an assistant professor. His research focuses mostly on obesity, inflammation, and fitness, and he will be talking about a very important and trendy topic, I think, today. So we're very excited to have you here. Thank you, Sean, for joining us.
Oh, my pleasure. Thank you for the invitation. And thanks for giving a little bit of my academic pedigree. I like to during my lectures give a little bit more background. So I'm gonna start off with that because it's probably obvious that I am not a native New Yorker by the way I'm speaking. So I'd like to give a little bit of background as to how I came to be a preventive cardiologist in New York, specifically because I actually grew up in Nebraska. Which is right here for New Yorkers who don't, and others who may not know exactly where it is, where I didn't really take Conestoga wagons to school, but certainly had relatives who may have, and which is called the beef state. So how did a boy that grew up in Nebraska, the beef state, come to be a preventive cardiologist in Nebraska? So the story of that actually begins even farther away. In Israel, actually, which is a place where I spent a summer back in college about 25 years ago now, actually, on a study abroad program in archaeology. And this study abroad program had a huge impact on me, both academically and professionally, obviously not to turn me into an archaeologist, but because of the exposure to the food that I had while in Israel, and specifically the Mediterranean diet, which is not at all similar to that of the beef state and the meat and potatoes that I grew up eating. But while in Israel, I really loved the food that I'd eaten there, and it was the first time that I had eaten a lot of certainly kind of like fresh olives for sure, and I'd never heard of hummus or couscous, tahini before, and really enjoyed the foods that I was eating in Israel. As a biology major at the time, I was particularly interested in the biological ramifications of the food, because at this time it was becoming well known, I think, even to the lay community, which a member of which I was, that the Mediterranean diet seemed to have some health benefits. And so again, as a budding biologist, I became interested in the underpinnings of those health benefits. And that was really the lasting impression on me from that study abroad. And when I returned back to the US, not to Nebraska, but to where I was going to college in North Carolina, which isn't the beef state, but which is, could be called the pulled pork barbecue state. Again, not at all, again, similar to the Mediterranean diet of Israel. I really missed those foods that I was eating. And so my only recompense was to learn how to cook them myself, because they weren't available in North Carolina at the time, similar to the lack of availability in Nebraska. And so during my last couple of years of undergraduate education, not only did I try to study nutrition within my biology studies, but I also pursued cooking with quite a passion, to the point where after I graduated from college, I actually worked as a personal chef for a year or so, while also working at the Duke Diet and Fitness Center, which was it remains an obesity treatment facility. I worked in the nutrition component of that facility, and so these experiences just contributed to my interest in nutrition and the role of obesity and in disease, and the potential preventative possibilities within that as a career. I plan to go to graduate school to further my studies of nutrition, but took another sojourn actually to a foreign country, to Japan, where I lived and worked for a couple years, and had multiple experiences abroad that convinced me that medicine actually would be a better place for my skills to be devoted. Nonetheless, I maintained a very strong interest in nutrition and its preventative capacity, and so did go to graduate school, but ended with a master's degree in Virginia, focusing on nutrition as well as exercise physiology. And with that background, actually returned to North Carolina, as you heard, to go to medical school at Duke. It seems I can't stay in place at Duke for the entirety of my education. And during my medical school career, I took a year off and went to the University of Pennsylvania, where I worked for a year doing research under the tutelage of this individual, Murdoch Riley, who is a preventive cardiologist and who's now actually across town here at Columbia, and who had a major influence on my career path. It was with Murdoch that I was first introduced to preventive cardiology specifically, but to research in HDL cholesterol, which is the topic I became very interested in. As you'll hear about, was really my first step into focused research of my own in obesity. After returning to Duke and graduating, as you heard, I was up in Boston for several years doing my internal medicine residency, and then arrived in New York at NYU about 11 and a half years ago. And I've been fortunate to find a place here at NYU that's really been supportive and allowed me to establish and start to expand a research program that's focused on the cardiovascular ramifications of obesity and specifically weight loss. And so that exists currently still. The goals of my translational research program is to understand how excess adiposity, excess body weight influence the development of cardiovascular diseases, as well as impact our traditional treatments and the potential resolution of cardiovascular disease. And it's through this research that I hope to gain insights and identify targets with which we might reduce cardiovascular risk, not only in people with obesity, but hopefully in all persons with cardiovascular disease or at risk for cardiovascular disease. And so whenever I speak to a physician audience, I like to remind people, or more often, honestly, make people aware for the first time that, while we now, atherosclerosis and cardiovascular disease prevention is really a realm of medications. It wasn't a medication, but actually a surgical procedure, and a GI surgical procedure specifically, that was one of the first interventions that we had to successfully reduce cardiovascular events. And this was a study that began in the 1970s at the University of Minnesota. The final outcomes of the study were published in 1990 in the New England Journal. The study being the Posh Trial, in which individuals who had had a recent myocardial infarction underwent a bypass surgery, but not a CABG, but actually a partial ileal bypass, bypassing a large portion of the small intestine, as you can see represented in this figure here, with the goal of reducing cardiovascular events, with the presumed mechanism of reducing hypercholesterolemia in these patients who, this being the 1970s and 80s, were in the pre-statin era. You can see from this primary figure from the trial that indeed performance of this surgery in these individuals resulted in a significant lowering of those who suffered an adverse cardiovascular event. And you can also see down here at the bottom that this is, largely likely due to the impact on circulating LDL cholesterol in these individuals who experienced, an almost 40% lowering of LDL cholesterol after undergoing this procedure. And so when I learned about this trial, I think early on in my fellowship, I was quite interested in it because as someone who'd had a longstanding interest in obesity, I was certainly well aware of the benefits of bariatric surgery and had seen the benefits of bariatric surgery in some of my patients from my cardiology clinic. And so as a budding researcher, I became curious as to the impact of our current bariatric surgical techniques, specifically on this outcome, the cholesterol outcomes of the procedures. An important question because the partial ileal bypass is really not something that's certainly not anything that's performed at all today in the bariatric realm, and also relevant because actually being significantly overweight or obese was an exclusion criteria for the Posh trial. So it was relatively a data-free zone. And so early on in my fellowship, I sought to answer that question: is what were the impacts of our currently performed bariatric surgical procedures on typical components of the typical lipid profile? And these data were published now about 6 years ago in the American Journal of Medicine, but you can see here, looking at the absolute change in LDL cholesterol, there's really quite a striking difference depending upon what surgical procedure you undergo, whether it's Roux-en-Y gastric bypass, a gastric banding, sleeve gastrectomy, or a biliopancreatic diversion, which is a rarely performed procedure, but one that actually bears some resemblance to the partial ileal bypass of the Posh trial. Additionally, there are also some, while maybe less significant, there are also variations in responses to HDL cholesterol across these groups. But I raise the fact that we're just looking specifically here in both LDL and HDL at the concentrations of the lipoproteins most relevant to HDL cholesterol, because as I had learned with my time at Murdoch, and then furthermore during my early time at NYU, where Ed Fisher had become my chief mentor, and who had and who continues to have a strong murine research program looking at atherosclerosis regression and the role of HDL within that process. I had learned that rather than the HDL quantity, it's more the quality likely of the HDL, the functional capacity of HDL that seems to be responsible for, or seems to be most important in regard to HDL's cardioprotective effects. And one of the primary mechanisms by which HDL is felt to be cardioprotective is in its participation in cholesterol efflux, so the removal of cholesterol from lipid-laden macrophages within atherosclerotic plaques in a process called cholesterol efflux, the removal of that cholesterol. And you can see here data from the Dallas Heart Study published by Anat Rohatgi now about 8 years ago, that those individuals whose HDL has the greatest capacity to remove cholesterol are at far less risk of experiencing an adverse cardiovascular event than those whose HDL is least functional in that regard. And so with my interest in HDL and HDL functionality, as well as obesity and bariatric surgery in mind, I set out to perform a clinical research study incorporating all of those interests. And the first step was to look at whether we could see an effect of obesity on HDL functionality in cholesterol efflux. And as others had shown before us, we did show that HDL from obese individuals exhibited a significant reduction in its ability to perform cholesterol efflux relative to that HDL from normal BMI individuals. The next question for me then was, well, if obese HDL is dysfunctional, what happens when someone loses a large amount of body weight? What happens to their HDL? And more specifically, does the type of way in which they lose weight via a different bariatric surgical procedures make a difference. And so we sought to ask whether there was a difference in impact of the two most commonly performed bariatric surgical procedures, a Roux-en-Y gastric bypass or a sleeve gastrectomy procedure. And we performed this study in collaboration with Manish Parikh, who directs bariatric surgery at Bellevue Hospital at NYU, and used a convenient sample of premenopausal Hispanic women who were undergoing one of these two procedures at Bellevue. We wanted to see the direct impact of the two surgical procedures, and so because of that, we excluded anyone that was taking a medication felt to potentially influence lipid levels or HDL functionality preoperatively, given the fact that after surgery, these patients undergo a lot of changes and in many cases, cessation of medical therapies following the bariatric surgery and subsequent weight loss. And you can see here the characteristics of our study population. Although it was not a randomized study, I would say that, overall the two groups were quite similar. There was only one statistically significant, although I'd argue not clinically significant difference between the two groups, that being age. Both of the groups were quite young, morbidly obese, but did not exhibit significant obesity-related comorbidities, again, due to our exclusion criteria at the beginning that wouldn't allow for them to have any complications that necessitated medical therapy. So our primary outcome time point of interest was 6 months, and you can see that regardless of surgical procedure, all subjects lost significant amounts of body weight, about 30% or so, by 6 months following surgery, exhibited substantial reductions in markers of systemic inflammation, improvements in insulin sensitivity, and reductions in circulating triglycerides. However, there were statistically significant differences in our primary outcome of interest, that being the change from baseline in cholesterol efflux capacity between the two procedures. And I'll be honest that the results were 180 degrees different than what I anticipated. The Roux-en-Y gastric bypass being the gold standard procedure, we actually saw a decrement in cholesterol efflux capacity at 6 months after that procedure, with a really null effect to slight improvement after sleeve gastrectomy. When you look at individual components of global cholesterol efflux, either that mediated by the ABCA1 receptor or not mediated by the ABCA1 receptor, you can see that it's really the difference in the non-ABCA1 mediated cholesterol efflux between the two procedures that's responsible for the difference overall. So again, this was quite surprising to us, and so we started to look into what might be predictive of the change in cholesterol efflux capacity following surgery. And when we looked at all of the available preoperative variables, there was only one that was actually predictive of the change in cholesterol efflux at 6 months, and that was the procedure that the individual had undergone, either the Roux-en-Y gastric bypass or the sleeve gastrectomy. When we looked at correlations with other variables that we had collected longitudinally in the study, we found that it was changes in ApoA1 in the circulation that were most predictive and most strongly associated with changes in cholesterol efflux capacity at 6 months. So I ventured to look at associations of ApoA1 with cholesterol efflux before and after surgery in the two surgical groups separated. And you can see here, on the left, I have baseline associations. On the right, we have 6-month associations. On the top, Roux-en-Y gastric bypass, on the bottom, sleeve gastrectomy. You can see preoperatively, there are relatively strong associations with ApoA1 concentration and ABCA1 cholesterol efflux regardless of the surgical procedure. And there's no statistical difference between the two. However, when you go out to 6 months, you can see that if anything, there's a strengthening of the positive association after sleeve gastrectomy, and maybe a weakening, certainly not an improvement after Roux-en-Y gastric bypass. And there is actually statistically significant difference between these two correlations now, suggesting that the characteristics of ApoA1 may be influenced by the type of surgery that an individual undergoes. And when thinking about why this might be the case, I went back to kind of the anatomy and looked at the pictures of the procedures, trying to think of why this might be the case. And I was reminded actually that, so while the primary site of synthesis of ApoA1 is actually the liver, there is actually a not small percent of ApoA1 that's synthesized within the intestine, specifically and specifically the duodenum, and that bypass of the duodenum, which is characteristic of the Roux-en-Y gastric bypass, may play a role in this change here. It may be that the source of your ApoA1 and in turn HDL may have, result in varying functionality of those HDL particles. And that was the hypothesis there that we put forth in the paper that we published a few years ago now. And I was excited to see this report that came out last year from Gwen Randolph's group, in which in mice, they actually showed that not cholesterol efflux capacity per se, but actually HDL's anti-inflammatory capacity. When they looked at systemic HDL versus HDL specifically coming from the intestines, the intestinal enteric-derived HDL, ApoA1, was more suppressive of LPS-induced inflammation, and that mice who had undergone a small bowel resection, in which there was no enterically derived ApoA1 and HDL, had far greater inflammatory response to an inflammatory stimulus, specifically LPS, than mice that underwent the sham procedure. So supportive, not directly of cholesterol efflux, but that HDL functionality broadly may rely somewhat on the location of synthesis of the HDL particles. And so, as I alluded to, there's cholesterol efflux capacity is one function of HDL, but there's also anti-inflammatory capacity that Gwen Randolph's group reported on. Those are just two of multiple potential beneficial and hypothesized roles of HDL in cardiovascular risk reduction, more of which are represented on this slide here, and some of which we're continuing to investigate within our cohort. So in collaboration with Alan Chait at the University of Washington, who has an assay that looks at adipocyte inflammation, we see that exposure to HDL from postoperative individuals is better able to suppress inflammatory response in adipocytes than is preoperative HDL. Although at least at this 12-month time point, which Alan investigated for us, there were no difference between the two procedures at that point. We're also have an ongoing collaboration with Uwe Tikke at the Karolinska Institute, who last year in Circulation published his endothelial cell inflammation assay, look in which he found that the HDL's anti-inflammatory capacity in endothelial cells was predictive in prospective cardiovascular events in a cohort of his patients. So we await results of that assay with bated breath. Hopefully we'll see a difference in the endothelial cell assay as well. And in parallel, we also hope to, if we see a difference in function at the endothelial cell level, but currently also looking just back at our cholesterol efflux data. We're trying to explain, what might be the case, why might the two different procedures have different effects. And so we're now investigating the cargo of HDL particles before and after each surgical procedure in collaboration with Thomas Weiser at the University of Washington, who did the proteomics, Thorsten Horneman at the University of Zurich, who performed the lipidomics, and then our bioinformatician here at NYU, Florencia Schlump, looking to see what changes before and after surgery. And Florencia sent me these figures yesterday, and as you can see, a lot changes, and some change up, some change down, some go up and then down, and there's a lot of data here that I'm very thankful to have a skilled bioinformatician helping me as we pursue things. But one of the first things that actually came out and that we noticed was that HDL after surgery tends to be particularly enriched for cholesterol ester 18-1. So this is oleate. And this happens at some point between surgery and 6 months after surgery. It remains elevated and enriched at both 6 and 12 months after surgery. And also of note, Ed Fisher has been looking at the lipidomics of HDL in his murine models of atherosclerosis regression, and also sees that HDL in his mouse models is enriched for cholesterol ester 18-1 or oleate. And when I first heard this, I was, oh, that's neat that the humans replicate the mice, or more appropriately, probably vice versa. But is that relevant? Well, I hope so. I certainly hope to make a case in a paper that will come out with shortly, because actually oleate is the second most abundant lipid in human plaques. And so this suggests to us that, perhaps this HDL is actually performing cholesterol efflux more effectively following bariatric surgery, at least specifically for this lipid. So as I was, performing that study, looking at the effects of bariatric surgery on HDL function, I took on the mentorship of another individual here at NYU, Jeffrey Berger, who's a platelet biologist. And together, he and Ed and I started kind of brainstorming ideas of work that I could take from my clinical studies and work to maybe bridge their two labs within the wet lab and on the bench. And thinking about that, we discussed that, platelets are now increasingly being recognized as immune cells rather than just, little pieces tiny pieces of megakaryocytes that act in hemostasis. They actually have a broad array of immune functions, although they're most frequently seen as inducers of inflammation. And so our thought was that, platelets are inflammatory. HDL is, good, the good cholesterol, and has some anti-inflammatory capacity. Is it possible that platelets may have a detrimental impact on HDL's function? And so we endeavored to investigate that by incorporating platelets into what we called the HDL anti-inflammatory assay. And this was an in vitro assay that took macrophages, exposed it to an inflammatory stimulus, LPS, so E. coli lipopolysaccharide, and then exposed these macrophages to HDL, which was presumably going to have an anti-inflammatory effect on the macrophages, and HDL that had or had not been incubated with platelets ahead of time. And then we would isolate the RNA from these macrophages, look at the expression of genes that would be indicative of an inflammatory or a pattern, and see, how it might be modulated by HDL and HDL that have been exposed to platelets. And I'll save you the description of about 2.5 years of work, repeating the assay over and over to try to convince Jeffrey and Ed that I actually knew what I was doing and I wasn't screwing things up and reversing plates and things overall, because HDL had no impact in this assay. Eventually quit putting it in, and it had no impact on the results. And not only did platelets not, stimulate inflammation, but when platelets were incorporated into the assay, they actually suppressed pro-inflammatory gene expression, which is commonly referred to as M1 polarization, a pro-inflammatory macrophage phenotype. So not only did HDL have no impact, but the platelets, which we saw completely as pro-inflammatory going in, had the exact opposite effect. And so, I was one day presenting my frustrating data at Ed's lab meeting, and he had just had a new postdoc arrive, Otto Weinstock, who's since actually left to start her own lab at the University of Chicago, who looked at my data and said, well, what about M2 polarization? So M1, as I alluded to, is a pro-inflammatory type phenotype. M2 is kind of the polar opposite, an anti-inflammatory, pro-resolving phenotype. And you can have one where you can, and so they're kind of separate phenotypes, and macrophages can switch from one to the other potentially. And so we modified our inflammatory assay to be to what we ended up calling an M2 polarization experiment, because the platelets were actually able to consistently induce that anti-inflammatory response. So we started looking at macrophages exposed to what I've abbreviated PR here, so platelet releasate, which is the supernatant appellated wash platelets, which is the experimental tool we ended up going to, because platelets have a very, do not survive outside the body for very long. And to protect my veins from being punctured multiple times each week, we would make platelet releasate from multiple donors in the lab and use it in experiments. But on exposure to platelet releasate, macrophages consistently had their phenotype altered into that of a pro-resolving state, indicated by the genes I've represented here: arginase 1, ABCA1, EDIL3, transglutaminase 2, with suppressed expression of pro-inflammatory type genes such as lipoprotein lipase, TNF alpha, IL6, and CCL2. And we saw this both in murine cell lines as well as human macrophages. And so, as I alluded to, it took a lot of convincing of Ed and Jeffrey that I was not really screwing things completely up in getting all these results. And, I was helped in this regard by my appraisal of the literature and bringing to them that, while platelets are typically seen as being pro-inflammatory, there has been a burgeoning literature on the platelets' role in curtailing inflammation, both in vitro and in vivo. And the results I was seeing weren't completely out of left field potentially. The question then, of course, becomes, well, what is it about platelets and platelet releasate that is responsible for these observations? And I'll say that we again endeavored for months and months to show that it was a protein, but the things that I did suggested it was certainly not. To the point where we ended up doing RNA sequencing analysis of macrophages that had been exposed to platelet releasate. And when you looked at the predicted factors upstream of upregulated genes in this RNA sequencing analysis, two really stood out to us. So one was a protein, PTGR4, but one, PGE2, prostaglandin E2, is obviously a lipid. And this protein, PTGR4, is actually one of the 4 receptors for PGE2. So this was attractive because it was a lipid and a lipid receptor that were predicted to be upstream of the genes that we were seeing influenced in our assays. And so, again, we question, is this something that we really want to pursue wholeheartedly? Because isn't PGE2 pro-inflammatory? Well, yes, in many situations, but again, there's an increasing literature to suggests that PGE2 can have inflammation resolving effects, and that the concentration and context of PGE2 exposure is really important. And so in collaboration with two individuals who were up in Boston at the time, Matt Spite and Brian Sansbury, we were able to show that we detected PGE2 in our platelet releaseate, and the concentration that we found it in was consistent with that had been previously reported to induce M2 macrophage polarization in the work of others. Furthermore, when we looked at PGE2 concentrations in our specimens and the degree to which they were able to modulate gene expression in our macrophages, we found that they were highly correlated, the gene expression changes and the concentrations of PGE2. We moved beyond that then to look at how we might be, if we could eliminate the effects of platelet releaseate on our macrophages, and found that when we exposed macrophages to an antagonist of the EP2 receptor, another prostaglandin E2 receptor, that there was no impact on gene expression, but that antagonism of the EP4 receptor, as suggested by our RNA-seq analysis, was able to suppress the responses that we saw. So further suggesting that it was PGE2 through binding to the EP4 receptor that was responsible for some of the changes in gene expression that we were seeing. So, this was exciting to us that, oh, well, wow, platelets were having these reproducible effects. They seem to be because of PGE2. And as someone who's been at this point for like 7 or 8 years, been surrounded by the concept of the holy grail of atherosclerotic plaque regression, this was pretty neat, because polarization, switching to an M2 phenotype, is something that Ed in his mouse models and other investigators have shown is necessary for the regression of atherosclerosis. That being said, while it may be necessary, it alone is not adequate. And there are other components and processes necessary to achieve atherosclerosis regression, one of which is cholesterol efflux, as I spoke about earlier in the talk, mediated by HDL or ApoA1 containing particles. So we look to see, well, maybe platelets can influence the ability of macrophages to participate in cholesterol efflux. And so we incorporated platelet releaseate into cholesterol efflux assays, the same type of assays that I had performed on our bariatric surgery samples. And as you can see here, platelet releaseate markedly upregulated cholesterol efflux, but then exposure of the platelet releaseate to a PGE2 antibody basically eliminated that increase in cholesterol efflux, as did exposure of the macrophages to an EP4 receptor, suggesting that a similar mechanism was responsible for the upregulation of cholesterol efflux in these macrophages. And then finally, another key component to atherosclerosis regression is phagocytosis. So the appropriate removal of apoptotic cells from atherosclerotic plaques, which results in atherosclerosis resolution and reduces plaque vulnerability to rupture. And so we incorporated platelet releaseate into assays of phagocytosis, in which you look at the capacity of macrophages to engulf apoptotic cells. In this case, in the experimental model, we use irradiated JURCAT, which are a lymphocyte cell line. And you can see here that platelet releaseate stimulated phagocytosis. The dying cells are in red, engulfed by the macrophages that are in green. And PGE2 antibody exposure basically nullified this effect, suggestive that platelet releaseate increased another component and function that is important to atherosclerosis regression, and that PGE2 within the platelet releaseate was key in this process. However, I'll bring to your attention that I don't present data on EP4 antagonism in the efferocytosis assay because it didn't work. It didn't cancel out the increase in efferocytosis. And furthermore, PGE2 itself had been investigated in assays of efferocytosis, and it hadn't been shown to influence it. And so we were kind of left scratching our heads as to, well, so what exactly is going on if our antibody works, but the antagonist of the receptor doesn't? And I was reminded by our collaborator up in Boston that PGE2 has been shown to be critical to the synthesis of other lipids that have actually been shown to be very important in inflammation resolution, atherosclerosis resolution, and induction of efferocytosis. Specifically, lipids derived from arachidonic acids such as PGE2, but called lipoxins, and from omega-3 fatty acids, the resolvent types of lipids. And so we questioned whether PGE2 present in the platelet releasate might regulate production of some other pro-resolving lipid mediators in macrophages. And so we exposed macrophages to platelet releasate with or without a PGE2 antibody, and then isolated the media 16 hours later from these macrophages, measuring levels of specialized pro-resolving lipid mediators in that media. And I'll save you the chore of looking through this heat map to basically say that there were very low levels of 15R, 15-epi-lipoxin A4 in the macrophage media initially. This was upregulated by macrophages exposed to platelet releasate, but this was not present in measurable quantities within the platelet releasate itself. And as you can see here, exposure of the platelet releasate to a PGE2 antibody basically eliminated the increased amount of 15R LXA4 in the macrophage media, suggesting that 15-epi-lipoxin A4 synthesis was induced by exposure to PGE2 in the platelet releasate. 15-epi-lipoxin A4 had been previously shown to increase efferocytosis in vivo, actually. So the takeaways from this process of investigation that was going on in parallel with my bariatric surgical study was that platelets can both possess anti-inflammatory and pro-resolving capacity in vivo. And these data added to the burgeoning literature that platelets might be not just pro-inflammatory immune effector cells, but have a pro-resolving anti-inflammatory capacity as well. Certainly, the limitations are not few. Key of which is that these data are in vitro and certainly would require validation in vivo. And I'll say that we have actually validated a number of these things in vivo, especially the most important outcome on atherosclerosis regression, and hope to publish those data soon. But key, again, to me as a clinician was that our platelet donors in all these experiments were healthy. And the question then remains is, well, what about platelets from non-healthy donors? And as someone who's very interested in obesity, the question was, well, what about the impact of obesity on platelets? Because I'd shown in other studies that I was performing at the time that certainly platelets from obese patients differ from those of lean patients, in particular of some of the more typical measures of platelet activity, looking at levels of activation as measured by levels of p-selectin. You can see that platelets from obese patients are far more active, but that this activity can be normalized by the performance of bariatric surgery. Likewise, the binding of platelets to neutrophils, which is seen to be a pro-inflammatory activity, is increased in obesity, and similarly reduced following bariatric surgery. So then we also did some additional experiments in vitro, looking at the impact of obese platelets on leukocytes, finding that the ability to suppress IL-6 expression, which we'd seen from platelets of lean individuals, was not possessed by platelets from obese individuals, nor was the ability to suppress netosis that we also had seen, which I didn't speak into in depth in lean individuals. So suggestive that platelets from obese individuals may not have as protective and pro-resolving a capacity as do platelets from lean individuals. And maybe the inflammatory capacity of platelets is context dependent. So we then set off on a number of kind of further investigations looking at the lipid content of platelets from obese and lean, both humans and mice, continuing in collaboration with our friends in Boston, and found that platelets from obese humans and mice contained reduced levels of a lipid that we had encountered in the study I just finished discussing, this 15R LXA4. In data from my first bariatric surgery study, we looked at the plasma of those patients before and after bariatric surgery and found that 15R LXA4 was substantially increased after surgery. So I started to become pretty interested in this lipid that seemed to be repeatedly popping up in our preliminary experimental analyses. And I had no idea, I'd never heard of this lipid 3 years ago. But as a clinician, became very, very intrigued when I learned that 15R LXA4 is also known as aspirin-triggered LXA4. So as a cardiologist, my ears really perked up when I heard that. So why is it called aspirin-triggered LXA4? Well, because it's actually synthesized in part by the activity of acetylated COX-2. So when COX-2 is acetylated, the capacity for COX-2 to synthesize 15R heat is increased, and it's 15R heat through the activity of 5-lipoxygenase that results in the synthesis of 15-epi-lipoxygenase 4. And this is something that we have, as I've just shown, is repeatedly shown to be deficient in obesity, but that we suggested may have pro-resolving and anti-inflammatory effects on leukocytes. And we are certainly not the first individuals to do this. There's been an extensive amount of preclinical data to suggest the role of aspirin-triggered LXA4 in the prevention and resolution of vascular disease, a slew of different mechanisms as listed here. And it was at this time that I was looking at these, getting my preliminary data, that about 4 years ago, as the data on this citation would suggest, that a meta-analysis in the Lancet was published that you may recall, that showed that when you looked at aspirin dose and the interaction of body weight and for the efficacy of primary prevention of cardiovascular disease, it came out that aspirin doses of 81 milligrams, low dose aspirin, were only efficacious at preventing cardiovascular events in those individuals with a body weight less than 70 kilograms, and were no longer efficacious as body weights increase. But in individuals with a body weight over 90 kilograms, doses at 325 milligrams and higher were efficacious. And the authors of this meta-analysis suggest or posit that this is due to inadequate platelet inhibition in higher weight individuals to aspirin doses of 100 milligrams or less. But the data to support that supposition are not strong and are in contrast with our own data, which we published earlier this year, in which we found that obese individuals who are absent diabetes do not exhibit any difference in platelet reactivity to low dose aspirin, regardless of body weight or BMI. So really calling into question whether it's due to a lack of platelet inhibition and COX-1 inhibition in platelets that's responsible for the body weight interaction there with efficacy and aspirin dose. So given that, those clinical data, clinical observations, and my own laboratory observations, it caused me to question, well, could it be another mechanism that aspirin's playing here? and specifically its role in COX-2 acetylation and the synthesis of 15-epilipoxygenase-4 that might be responsible for the observations in that meta-analysis. Specifically, could inadequate COX-2 acetylation and submaximal 15-RLXA4 synthesis be responsible for ineffective cardiovascular event prevention with increasing body weight? And we're now actually pursuing that question. This is the focus of an R01 I was fortunate to get about 1.5 years ago, in which we have two aims. The first is a randomized trial that looks to address this hypothesis, is that at low body weights, regardless of aspirin dose, you get maximal synthesis of 15-epilipoxygenase-4. But that as body weight increases, the synthesis of 15-epilipoxygenase-4 to low dose aspirin falls off, where it's sustained at a high dose of aspirin. And we're assessing this question using a randomized controlled crossover design study in which individuals come in and undergo a placebo run-in, then take one of 2 aspirin doses, undergo a washout, and then take the other dose of aspirin. And we draw blood prior to and following each visit to assess the primary outcome measure of plasma epilipoxygenase-4, as well as a whole slew of other variables there that we're assessing complementarily. And just to kind of tell you where we're at, we're targeting at least 72 subjects across a wide range of body weights, with equal representation of men and women. As of today, we have 19 subjects who've completed all 4 visits, and we've analyzed data on the first 10, suggesting that aspirin seems to stimulate synthesis of 15-epilipoxygenase-4, so that we're happy. Seems to be working. We don't know I don't know what dose anyone's getting, because that is blinded. We'll see at the end, but I'm heartened by that our assay seems to work and the intervention seems to work. Aim 2 of this study is actually a nested case control study within the ischemia trial to see if plasma levels of 15-epilipoxygenase-4 at the time of enrollment into ischemia can be predictive of major adverse cardiovascular events in this population with significant but stable coronary artery disease. And so with that, I show you Team Discover. That's the name of our randomized trial looking at body weight, the interaction of body weight and aspirin dose on 15-epilipoxygenase-4. And thanks for your attention and for the invitation here today. I think I have 2 minutes for questions, but I'm happy to stay longer if you can host longer. But my email address is here if you want to reach out with any comments or questions. I'd be happy to discuss them at length with you. And thanks again for the opportunity.
Thank you very much, Sean, for that. That was an amazing lecture, and I love the translation aspect that you put into it. So everyone, for your questions, you can put them in the QA, and we'll see also the CME code now there by Millie. I can start with a question. I think this is a very, from the research, but also for us that we see patients, when we evaluate the risk of patients, when we're seeing them, obesity is usually not there in the scores that we look at, and it's not part of the evaluation. We kind of see it as from our experience, but it's hard to put into scores. So the question where I'm going for is there any limit in the BMI, or is there any healthy fat that you can have of a healthy amount of overweight, or the inflammation increases right after a certain body weight?
So that's a good question, because certainly, the obesity paradox has been reproduced in many different situations, which certainly makes one wonder. I guess a couple of things that I've become convinced of in my both clinical as well as research experience is that I remain convinced, although we haven't proven it yet, but hope to shortly, that the duration of obesity really is important. And you know, it's kind of like the LDL exposure, kind of the area under the curve, and the duration is really important, because when I started off to do my bariatric surgery study, lots of my colleagues were like, "How are you ever going to find morbidly obese people who aren't on these medications?" And I was like, "Well, you get them early enough, and they haven't succumbed to that." And so, like, the duration is important, and if we can intervene sooner, that is likely helpful. I guess going beyond that, even in the setting of obesity, having healthy lifestyles and I guess modulated risk factors within obesity can certainly help reduce the associated risk. So the concept of fat fit is beneficial. I mean, would slim or normal fit be better? Yeah, yes, likely, but there are a great number of obese individuals who are cardiovascularly fit who overall are at lower risk than the sedentary normal BMI or low BMI individual.
Thank you. That's a very good point. We have a very good question, I think, from our fellows. They ask, is there any indication that inflammation in obese patients is different pre-bariatric surgery and after surgery if they gain the weight back?
So that's a really good question, and actually one we're actively trying to investigate now, because the concept of immune memory is very prominent, and the thought process that exposure to chronic inflammation, exposure to excess adipose and the cytokines released by such and other circulating factors may actually influence epigenetic changes of the DNA, which may influence inflammation that occurs post weight loss, and then certainly in the setting of weight regain. So to answer that question, we don't know yet. I would not at all be surprised if it does. So Ed, as I mentioned multiple times, and Ada, his postdoc, who's now at the University of Chicago, have done a lot of mouse research on this, and we'll have a few publications coming out in the next year to suggest that there's probably a slight difference in mice, not a huge one. We're starting to look at that in human samples that are harder to come by, because you don't know who's going to regain weight versus, so you need to get a lot of pre-op and post-op samples in both situations to catch those people. But in long answer, I guess in short answer, even though I've spoken a long time, I wouldn't at all be surprised, because immune memory, I think, has been demonstrated in hyperlipidemia in humans, and I wouldn't be at all surprised in obesity.
Thank you very much. A great question that is somewhat related from Anna Bornik. Is there any data on the effect of the GLP-1 receptor agonist on change in cholesterol efflux profile? Is it just a weight, or you think this could extend to the new medications that could do a similar effect?
Yeah, so there is some evidence to suggest the GLP-1 receptor agonists improve cholesterol efflux. Those in vivo data make it hard to attribute the impact specifically to the medication, because generally speaking, weight loss tends to improve, especially once weight loss has stabilized. It tends to improve cholesterol efflux capacity. So it's probably there's probably a benefit, broadly speaking, of weight loss on cholesterol efflux capacity. Whether GLP-1 receptor agonists have an additive or complementary effect to that isn't yet clear.
Okay, thank you. Thank you. Very, very good lecture. We're very excited about reading your research in the near future, and hopefully more collaborations as well.
I look forward to it as well. Thanks again for the invitation.
Take care. Bye-bye.
Bye.